So most of this video will complement the background review that's available in your Blackboard module. Over the next couple of lectures, we are going to review the difference between Mendelian traits and human traits, because ultimately, after this unit, you will understand the complexity of human genetics. And it's not what we anticipated. So just to review, Mendelian traits are easily distinguishable, constant throughout life, controlled by one gene, right? One gene to one protein. Traits are dimorphic, so green versus yellow. Traits are consistent, have consistent dominant and recessive patterns. They are inherited independently, and the inheritance determines genotype. So in pea plants, or in traits that are considered to be Mendelian traits, the genotype equals the phenotype. And we have tried our best to apply this to human genetics, but it just turns out that we are a little bit more complex. So our traits are difficult to measure, the ones that we're really interested in, right? Like, who cares if you have a certain shape of like fingernails or whatever, right? We care about more complex traits such as obesity or height or criminality, right? And that's difficult to measure. Our traits change over time. Traits are influenced by multiple genes and environmental factors, so we don't have simple one gene, one protein traits. Most of our traits are continuous. Most genes have more than 2 allelic variants, and that'll be really important to understand, that we don't just have a dominant and a recessive. And then to build on that, dominance is less predictable. One gene can influence multiple traits, and the environment can change your genotype. So we're talking about like the environment mutating our genes, or even turning off our genes. Gene expression. So in humans, the genotype influences, but it does not determine phenotype. Important to know. Okay, moving on. So I do want you to know some basic anatomy, the difference between a gene, a chromosome, DNA. Histones will become important later on, but you'll understand what those are as we go through them. And please watch any of the content that's available in the background section of this module. So it's important to know that most of our DNA does not code for protein. So we consider genes the specific nucleotide sequence that codes for protein. And almost 99% of our DNA are not coding for proteins. We were pretty disappointed after the Human Genome Project to find that humans only had 20,000 genes. So corn, for example, has more genes than we do. So, like, think of that next time you're at a barbecue. But that means that something else is missing, right? Like, the number of genes doesn't explain the complexity. So what is all of that non-coding DNA? So at one point it was termed junk DNA, and then we also have the epigenome, which is are the structures on top or beside our DNA, and we'll talk more about that later. But the junk DNA, what are those? So you have satellite DNA that really help with structure. You have telomeres, which are really important in terms of aging and disease. And so remember, these telomeres are at the very end, and they shorten with every duplication of the DNA. But you do have an enzyme, which is a protein called telomerase, right? And telomerase rebuilds the ends of our telomeres, and that can be overactive in cancer. So you have this balance of telomere deterioration and telomerase building it up. And then within the gene, the protein coding sequence, you have introns and exons. So you have bits and pieces that will eventually be spliced out. And then you have non-coding RNA genes. So you have DNA that also codes for RNA that doesn't eventually become a protein, right? Like that's where you get the genes for your tRNA. And then you have these regulatory sequences, and these will be really important to know about. So you have sequences that are either upstream or downstream from the gene, from the protein coding sequence, and these are involved in transcription. And so these include the promotion, the promoters, the enhancers, the silencers, and so these are like these switches that turn gene transcription on and off. So this should be familiar with you. This is the central dogma. DNA is transcribed into RNA, and that's translated into proteins. And this is this does not explain how things work in humans, actually. We are way more complex. And so this video is available in the Blackboard module, but this is what we are building on, not what we are going to be testing over. So mechanisms that affect gene expression, because we are really interested in the gene expression, right? We're interested in the trait or the protein or the product, what comes out, right? Because the sequence only tells you so much. And so, as can be predicted, there are genetic changes that can affect the gene expression. So if you have mutations, right, that's easy to imagine. You have a mutation, yes, that will affect your gene expression. You have some structural damage, so those are like your deletions and insertions that change the structure of the chromosome. You have mosaicism, which we'll talk about later, and then you have epigenetic mechanisms. So epigenetic, again, beside or in addition to the gene. And so we will talk about these at length. But some of the mechanisms involved in this are DNA methylation, histone modification, and using microRNAs. So one huge point to understand is that genes don't decide when a protein is made. They are not autonomous entities that know all the creation of life and da-da-da-da. They don't determine anything. They're tools that can be used in the right environment. So it's really the environment that regulates gene expression, and you really can't prioritize one or another because they work together. The environment, and whether that environment means the intracellular, the adjoining cell, or if it's a signal from far away, like a hormone, or a signal from outside, like sunlight, or like, reactive oxygen species, right? So signals from anywhere that turn genes on or off. So for example, in a high pollution environment, our bodies can turn on detoxification enzymes to make more antioxidants, right? Okay, so genes determine nothing. It's the environment that determine which genes are turned on and off. So one way we regulate gene expression is by transcription factors, and these are switches. So these are those upstream nucleotide sequences that turn genes on and off. And there are different ways that we can affect the transcription factors, but this is just a visual depiction of what we're talking about. So you have the promoter sequence, right? And RNA polymerase binds to that promoter sequence, and when that happens, okay, we can start transcribing a gene. But before that happens, you actually have to have the binding of all these different transcription factors, but they also have to bind to certain enhancer sites. And so it's only when all of this complex is bound that you can start gene transcription. So if, for instance, one of these sites is methylated, then you don't start gene transcription. Gene expression is impacted. The other thing to realize is that genes work in networks. So multiple switches may activate a single gene, and a single switch may activate multiple genes. And, this is kind of a depiction of the complexity, right? So this increases our complexity of how our cells can react to signals from the environment. So say we are hurt, we might activate transcription for some of those inflammatory processes, right, to occur. And for cell division to occur. But say we are in a nutrient-poor environment, then we also have messages that are telling our cells not to divide. And so just think of all of these different switches kind of like different signals communicating to the same gene to turn it on or off. But the main point is that these things work in networks. And then you have regulation by epigenetic mechanisms. So let's see. So you can have a methyl group. You all remember what methyl groups are from chemistry. You can have that. Just bound to the gene or bound to the switch, and then it's not. You can't transcribe that portion. You can have DNA wrapped around these proteins called histones, and then they are inaccessible to transcription. And then you can also have microRNAs, which is not in this photo. Okay, so genes work in networks, right? So one switch can turn on or off multiple genes, and then multiple switches can turn on or off one gene. Okay, so moving forward after transcription, we also have something called alternative splicing. And so what this means is one gene does not code for one protein. We have a setup where one gene codes for multiple proteins, and that is why we can have only 20,000 different genes, but so many more different expressions of that gene or different proteins that come out because we have alternative splicing. So basically, that nucleotide sequence that is initially transcribed is then split up into various different ways. And think of the ways that we have an alphabet with a certain amount of letters, right? But you can mix up that alphabet so many different ways to make different words. And so that's kind of where alternative splicing comes in. And this also allows us to respond to our environment signals in different ways. So now we can move beyond the central dogma because it's not DNA is transcribed into RNA and that's translated into a protein, right? We have epigenetic changes that affect this. We have a DNA structure that affects this. We have different protein interactions that affect this. We have microRNA, which we haven't really talked about, that can affect this. And then that's transcribed into pre-RNA, and that pre-RNA is processed through alternative splicing into different, mRNAs. And then those are translated into proteins. And then you have additional factors that come into play with the structure of the protein. But basically what this means is that it's a little bit more complex than Mendel's pea plants.